So when I was growing up, I actually wanted to be a physician. When I volunteered in the ER, I soon realized that I was only going to be able to help one person at one time. And I thought, there's got to be more that I can do for more people. So when I started out in my research career, I was very interested in the interface between biology and chemistry. I could bring those two worlds effectively together and do something more. Innovation is driven by people who don't look behind themselves at what others have done, but they ultimately have a resolute focus on where they're going and what the next steps are. Developing a standalone translational research enterprise requires a village made up of constituents that bring multiple expertise to the table, because we need the scientific expertise to marry with the clinical expertise to define the problems and define what can and what can't be done in a feasible fashion. I'll give you a metric. Many of the research projects that we've been doing up until now, we've been attempting to access clinical specimens from national cooperative groups or other stakeholders that have biospecimens available from biobanking programs. We had a study design that dictated us needing 300 uterine cancer specimens. When our histopathology group and our consulting pathologist reviewed these, only about 40% of these specimens were viable for cancer research. And so what we had to do then was get back in line, request another set of samples, and this went on essentially for 5 years. And so you can appreciate that that delay of 5 years is a real measurable delay in research. So I think the most critical thing is the ability to access highly qualified patient specimens. We're approved by the College of American Pathologists. That means that the specimens in our bank are highly qualified, and along with that, we spend a lot of time collecting data points from these patients. The best advice that I can give somebody who is venturing into the field of LC-MS and proteomics is to have definable hypotheses, incorporate controls, and incorporate replicate measurements to ultimately uncover the complexity of the proteome. But I would remind people that it's much more complicated than we previously thought. We develop partnerships with industrial partners who are engaged, who want to be seeing their technology being used in a way that is translationally relevant. And I think Thermo Fisher ultimately represents really an optimal partner, because they are focused not just on rapidly advancing technology, but they're also very much sensitive to the needs before anything gets to that technology.